the special case of biofilms. And so biofilms are, oh, that's weird. There we go. Everywhere. Biofilms are everywhere is the short answer. It seems the more they study that biofilms, and then especially the more they take samples from out in the wild and look at how microbes are living their life outside of a lab, it seems like the biofilm strategy is super common and that pretty much any microbe can do this and often chooses to do this. And so, we talked about the glycocalyx, capsule and slime layers that bacteria make. So they synthesize these polysaccharides and proteins, and then they excrete it out and kind of coat their whole cell surface with these layers. And then if you have a whole bunch of microbes all doing that all in one space, we call it a biofilm. And the classic example I gave you the other week or so was when you wake up in the morning, if you've had a solid night's sleep and you feel that fuzzy goo across your teeth, that's biofilm. And we're going to get into human microbe interactions in a little while, but I'm reading a book right now on human microbe interactions, and the dental plaque that the technician scrapes off your teeth is the second highest body site in terms of microbe number. Your gut is the first one, your gastrointestinal tract. Dental plaque is the second highest thing. I was shocked to hear that. So I'm gonna see if I can find out more information on that so we can talk a little bit more about it. Here you're looking at a bunch of cells under the microscope in a biofilm. This is figure A down here looks like a lot of cyanobacteria, maybe some algae mixed in and a bunch of other bacteria, looks like in an aquatic environment. And then of course teeth, and then it can get really significant if you aren't having good oral hygiene and can have lots of, this scraping, they took a scraping and this is what they found for the teeth surface. So lots and lots of microbes hanging out in dental plaque. So they think it's kind of a natural state. It can be very good from the perspective of the microbe, and it can be not so good for other situations. The dental plaque idea, obviously. Even on medical devices, this is a big problem in hospital settings. If you think about having, tubes put in you, intubation tubes, catheter tubes, things that, especially catheter tubes, because they're so small, it's hard to sterilize those adequately, and then they're super easy to get colonized once they're inserted. So you have these challenging issues where you have to have very strict protocols on making sure that any kind of medical devices are successfully sterilized. Hopefully they don't have the type of autoclaves we currently have at hospitals. I don't know, our autoclaves, we have two major autoclaves, and the left one is down and out right now. We can't even use it. And the other one fails more frequently than it should, considering they're fairly new. But anyway, so hopefully hospitals have better autoclave systems. But basically, when you look at microbes out in nature, living their life, it's usually a mixture of species that are in that biofilm. It's very rare that you would see outside of the lab environment where they're doing, actual research on it. It's very rare that you just see one species biofilm. So biofilms, let's think about this idea. Most of you are familiar with hot tubs, right? Or jacuzzis. Maybe less so with sauna, and it's sauna, not sauna, right? So if you're not so familiar with the sauna idea, you go in, you sit down, you usually put a towel down to be polite, and it's heat that comes either from a wood stove or an electric stove, and it heats up the room quite hot, around 160 to 180 degrees Fahrenheit. And then you can splash water on the rocks to create some steam. but it's a relatively dry experience compared to immersed in water with a hot tub, right? And then if you've ever had to like clean a hot tub or work with a hot tub, you know that there's pipes leading in to, you know, pipe in the water after you're done cleaning it. And depending on the hot tub, you can fit a lot of people apparently into the hot tub, so there's another thing to consider. So let me ask you another Google question. Thinking a little bit about these two scenarios. Whoops, where'd it go? Try this one. Like you're in the dead zone over there. That's okay. Oops. Oh, here it is. All right. Let me go back. Oh, that's weird. Why it's doing that. All right, let's try that again. Okay. So what did I ask? How did I phrase it? Lower case of biofilm?
Which would have less chance.
Which would have less chance of biofilm
Build up and persistence.
build up in persistence. Okay, so hot tub or sauna? Think about biofilms and microbes, happiness. Like, where would they be happier? Sauna wins in terms of less biofilm, less persistence. So you have to kind of flip it and think about the microbes' lifestyle and what they like. Water, of course, they have to thrive. Any living organism has to have water. So here's a great source of water. And basically, you're taking a bath with a bunch of strangers if you go to a resort and, you know, pop yourself into a hot tub. I mean, essentially, you're not like you can just have your own fresh little water around you, you know, it's all mixing. So, you have all that going on. So lots of skin sloughing, microbe sloughing off of the bodies in the water, watery environment, even with the chemicals. I know they put chemicals in there to try to keep down numbers. So you might have been thinking about that and answered hot tub. But there's been some interesting case studies that have occurred where people, especially at resort areas where the hot tubs are not cleaned adequately. There was one case study where a young college-age male went temporarily blind in one of his eyes because he was in a hot tub and, you know, it produces those aerosols, right? You know, it's bubbling along, it feels great. There's no doubt about that, to have that bubbly water. But it creates all these little tiny aerosols that can get up onto your face, into your eye, in your respiratory tract. And apparently he had his hands in the water and then he went to rub his eye and he scratched his cornea just a little bit with his fingernail and it had Pseudomonas. And so he introduced Pseudomonas from the water into his eye and he got really badly infected, went to the doctor. Luckily, it was a Pseudomonas that was sensitive to antibiotics, so he recovered his sight, but they trapped it down to the hot tub. and were able to diagnose that, you know, the epidemiologist, that that was the likely culprit. And so then there was another case study where two women in their 70s, I believe, didn't even go in the hot tub. They were just sitting along the edge and dangling their legs in a hot tub at a hotel resort. And then they both contracted herpes, genital herpes. And the doctor's like, "Were you having lots of sex on your vacation?" And they're like, "No, you know, we were just having fun doing other stuff." And what they tracked it to, the epidemiologist found out that where they were sitting on the hot tub, because you know, the water splashing, they were sitting in a wet area on the hot tub, and it turned out that another person, a young male, sat there right before them, and he had an active genital herpes outbreak. Apparently sat there, got out, and then they sat there. And so, lovely stories, I know. And I'm sorry if spring break is around the corner and you're going hot tubbing. But I actually went hot tubbing at a resort once when my youngest son was like 5, and he was standing there. We were at Tundra Lodge in Green Bay, you know, that resort. And it's a great water park, but he was like shivering and just shaking. He's like, "Can we go into the hot tub?" And I, you know, all this is going through my head, and I'm like, oh, how do you say no to a shivering 5-year-old, you know, your son? So I'm like, okay. And luckily nothing happened, but yeah, I had to turn my microbe brain off and just meditate while I was in there until he warmed up, and then I'm like, okay, let's go. So just be careful. I want you all to be happy and healthy. I don't want you to have any issues, so be careful about hot tubs. I still go in hot tubs if I know whose hot tub it is, like my sister has one, so I'll go in hers. Because we share probably a lot of the same germs anyways and microbes, and I know they take good care. The other issue too, when, especially at resorts or in larger hotels where they have hot tubs that are frequently used, the other trick that they have to know is that when they drain the water out, they have to think about the water pipe. That when they go to turn the water back on and the water comes through that pipe, if they can clean the basin of the tub where people sit and get that totally sterile, great. But what about the pipe? The pipe has biofilms of microbes. And so unless they treat that section too, they're going to, when they turn the water on, have a flush of water with microbes coming with it. And so it's a lot more challenging to clean successfully a hot tub system than it is a sauna. Sauna is very low risk, especially if you bring your own towel and sit on it. It's a dry heat, hot heat. So, have I convinced you? So sauna is a better option, and saunas feel great too. So definitely worth it. So thumbs up for saunas. Okay. So that's my plug. Even I am Finnish, from Finnish descent. So obviously I grew up with this idea, but lots of health benefits outside of a safer bet in terms of less biofilms. Saunas are very healthy, help with your cardiovascular system. Okay, so they now suspect that pretty much any biofilm that's getting formed is due to several processes that seem to have to happen in a certain order. And one of them is quorum sensing, which I'm going to be talking about soon anyways. But basically this is the microbes' ability to communicate and assess their environment around them. And so more and more, when they look at these mixed species biofilms in particular, they're seeing that is heavily used. The other key thing, they need to like attach somewhere, whether it's your teeth or the pipe, water pipe, they will start to attach. And then as they're doing that, they're quorum sensing, and then they will shift into the biofilm lifestyle. And so you tend to get these cells all kind of congregating and in these groups or consortiums, and then eventually they'll kick into the biofilm process where they start to create all those polysaccharides and proteins and excrete that all out. And then if all the cells are doing that, then they can form a nice biofilm. They do all kinds of really cool studies with these like miniature biofilm systems in labs, and then they have these really powerful video cameras and camera systems. And it does look like kind of like, I think about downtown Chicago or something like that, where you have these skyscrapers and you have these through, streets. And biofilms tend to be like that. You have parts that are built up, then you have like hollow areas where fluid and nutrients can come through it. Kind of like a downtown Chicago is usually what I think of. And so it can be super beneficial for the bacteria or the microbes to get into this lifestyle because it creates this area, especially if they're like, oh, this is a really rich area of nutrients. I'm going to settle here for a while, form a biofilm and use up all these nutrients and have a good life in this spot. So they will do that. But then if those nutrients become scarce, or if there's some kind of toxin that comes into the system, maybe, then they're not permanently in the biofilm. They can come out of it and disperse and find a new habitat. So it's not like it's a permanent thing. It's very much, I'll go into the biofilm because it's advantageous at the moment, but then they can move out and disperse. So often in natural settings, there's the hypothesis that they would certainly enter into biofilm around a rich source of nutrients, because then it's like setting up camp and being able to take advantage of all those nutrients. And then biofilms create the situation where you have lots of cells all really close to each other, near each other. It's a real neighborhood of microbes. And so that allows for horizontal gene transfer. So they do see that happening a lot in biofilms, where it's kind of a hot spot for horizontal gene transfer going on amongst all the different bacteria. So here is trying to show the general process where if you imagine a river with rocks or logs, and the bacteria are like, oh, I like this log, it has a lot of carbohydrate here. I'm going to attach to the log surface, and then you get enough of them doing that all in one spot, and they're like, oh, okay. And they're quorum sensing, they're figuring it out, and then they start making this biofilm. And then this depiction, they're trying to show that you can have like height to the biofilm, like the skyscraper idea, but you can have like through ways that are hollow, where you have the river water rushing through parts of it more easily. The picture, like with all the green, that was a stained bacteria. Again, they're trying to show that there's this definite three-dimensional build, like a downtown city with the buildings. This might be a little bit easier to see what's going on. So that, again, they're trying to show the three-dimensional aspect of the biofilm. And so you can imagine that this could be useful for the microbes. If this is a nice patch of nutrients, they can stay here for a while, stay put because of all the biofilm, and they can do horizontal gene transfer. They can, maybe they need to coordinate their behavior. And so we'll talk about quorum sensing, how they can do quorum sensing and do that. Maybe they are, oh, here's another picture too, where they're trying to show the three-dimensional, that's a little blurry, three-dimensional aspect where you have these channels. That's the word I'm going for. These look like water channels, and then down low you can see the water channels from a different perspective. So here, they're really getting into some nice sophisticated study tools in the lab to like allow for these natural biofilms to develop. And it can be very advantageous for the microbes. So whether or not they're trying to attach to your teeth all night long and get access to nutrients in your mouth, or the water pipes in your home, for example, or in the city. Medical equipment, unfortunately, not so good, but it's a good strategy to allow them to persist in an environment where things are going really well for them. And then when things start to get stressful, they can just pull up the tent stakes, so to speak, and move on. So here, this is a mixed biofilm, two different bacteria. I believe the red is a bacillus. No, Bacillus is green and Staph aureus is red. So you've worked with both of these, B. subtilis and Staphylococcus aureus mixed biofilm. And here it says the presence of B. subtilis protects Staph aureus from a disinfectant treatment. So this is a piece of medical equipment where they were running a test to see what would happen if you have a mixed biofilm. And, these are two, B. subtilis and Staph aureus are very common on human skin. And so they were looking to see what happens if medical equipment gets contaminated with them. And they actually found that the bacillus was helping protect Staph aureus from the disinfectant. And so that it was persisting. And so these types of questions, it's like, how worried do we have to be about biofilm? And especially if you have all that sticky goo, the biofilm, it's hard to disinfect that off, to get rid of that, to successfully kill the cells. You have to get drugs that can permeate that biofilm and really effectively kill the microbes. And so it can be very protective to them. If they're in your body, in a wound, for example, and your phagocytes try to engulf biofilm bacteria, good luck. I mean, that's just, they can't. If the biofilm is too extensive, your phagocytes won't really be able to ingest the bacteria successfully. So against the immune system, it could be tough. And it certainly helps you deal with dehydration in your environment. So if your environment starts to get drought prone, if you have a biofilm that you're living in, you're good. Certainly antibiotics have a much harder time diffusing through a biofilm. So if you are sick with a pathogen that produces biofilm, it'd be much harder to treat. And the microbes that I've worked with that have been directly isolated from patients that are sick, almost every single one that I was working with had really nice biofilms, just really extravagant biofilms when I would do streak plates of these pathogens. So it's a common lifestyle that pathogens will pick, so to speak, because it gives them a lot of advantages. So here is this idea of, okay, so sometimes we don't want biofilms, like on medical equipment. How can we get rid of them? They're actually using viruses, phages, bacteriophages. And so they're actually putting so-called smart bombs inside the phage and trying to see if they introduce the phage into the biofilm, if the phage will go and find the bacteria and then insert the so-called smart bomb to kill off. In this case, it was Enterococcus faecalis. I think you might have worked with that one already in lab. You can't really see maybe in the light here, but this is before treatment. You can see a huge biofilm, and I don't know if you can see these little green streaks here, but mostly black, showing just minimal green there. And so the phage effectively killed off the Enterococcus. And so these types of things they're really interested in because fighting biofilms, it's really hard to get at the cells in the biofilm in order to kill them. So here, this is another one where they're attacking anti-biofilm peptides and looking at that on Pseudomonas. So green is live, red is dead. And so you can see before or no peptide treatment. And then the figure B is afterwards. So in this case, they stopped the biofilm production as well. So it's a big concern, especially when you think about biofilms in humans, which there's plenty of that. The bacteria could gain some resistance, potentially. That's why I think right now, the use of phages is a little bit more attractive for that, from that perspective, because I think there's, thinking that could be a more effective way where resistance wouldn't build up so easily. That's the last I read. I'm assuming that's still the case, but so yeah, they're pretty excited about phages, the use of phages to attack the bacteria in the biofilm. So for people, especially if you think about like cystic fibrosis and things like that, where you get quite the biofilm production in lung tissue, due to pathogens, that this is a huge problem. And so they're trying, different strategies to limit this kind of biofilm in the human body. So I'm going to come back to this idea of quorum quenching, but I just wanted to introduce it right now. So this idea that as the bacteria are hanging out in close quarters, they're really, jammed in there in the biofilm, and they can have really large populations. So they do communicate with each other through this process called quorum sensing, which I'll get into. And so kind of the opposite of that is, okay, if they're quorum sensing and that's a strategy they're using to be successful biofilm lifestyle, that's what they're doing, then we got to stop the quorum sensing. Can we do that? And so they nicknamed it quorum quenching, which just means you break the quorum sensing process, you stop it. And that has some indication that it could work, but it's still young in the research phase of this. But they're really starting to consider this heavily, this quorum quenching, because we know all these antibiotics, that are out there right now. There's so many microbes that are resistant to those antibiotics and they're using quorum sensing to gain resistance in large part. So if we can do quorum quenching, they shouldn't build resistance against that. And so quorum quenching is really starting to be looked at heavily for a way to disrupt biofilm formation. Just stop it from happening. If you could stop it from happening, that would be the best situation, especially when you think about patients with cystic fibrosis or something like that. You can watch the biofilm video on your own. So I'm going to come back to some of these ideas again when we get into quorum sensing, but thinking about the natural life cycle of like typical bacteria, whether they're pathogens in the human body or if they're just living their life out in the wild, so to speak. Biofilm formation is way more common than they first hypothesized. And so they're really getting into figuring that all out. So another process. So, if you have these microbes and they're hanging out, and let's say they do form a biofilm, and then the nutrients, I gotta go out of this, hang on just a sec. They set up camp in an area where there's really good nutrients and they want to stay there for a while, so they form a nice biofilm. Well, then eventually there could be lack of nutrients. So they go through some kind of stress event in their life where like, okay, I'm running out of water, I'm running out of nutrients, or maybe there's some toxins around me. I need to try to deal with that. And, we've talked about maybe they could form spores. I mean, that would be one process, but there can be other options and lifestyles that bacteria and fungi could take if they need to. So we have talked about endospores, so I'll just bring it up briefly, but that is a strategy and it is a form of differentiation. I'll talk to you about heterocysts and fruiting bodies, and then some aerial hyphae ideas. And so here it's looking at the idea that they can shift their life and growth form and growth phases in response to the environment where, they might be facing tough times of some sort. We've already talked about endospores, so I'm just putting this here to remind you that that is definitely a very good option. If you're gram-positive bacteria and things get tough, this could be an option that could let you persist in the spore state for thousands of years, actually. So the endospores kind of top the list in terms of just incredibly resistant and long-term options. Here's another option with the formation of heterocysts, which you can see like the regular vegetative cells here of the cyanobacteria. And then you can see some like brighter cells under the microscope that look, a little bit bigger, a little bit different. So these are, Anabina is a type of cyanobacteria. And those heterocysts are an option. They can build, essentially, it's a specialized cell. So if they're in an area where they're like, I'm running out of nitrogen, I don't have any nitrogen around me, I need nitrogen, they can shift and some of their cells become heterocysts. And then in that heterocyst, they'll start to fix nitrogen. And so that's the idea where they're gonna take nitrogen gas from the atmosphere. And so they'll take N2 from the atmosphere and they'll fix it. And when they fix it, it fixes into ammonia, NH3. So N2 to NH3, and then the ammonia usually gets quickly put into amino acid synthesis. So it's just a way for them to switch their growth phase so that they can do the job of, making their own usable nitrogen source out of dinitrogen from the atmosphere. So the trick here, they have to spatially segregate it because Anabina is an aerobic organism. They're doing photosynthesis, so they're making oxygen in that process. They like oxygen. And yet, when you fix nitrogen, you have to have a pretty anaerobic environment. So what they do is they create these heterocysts and make it anaerobic. So all the rest of the cells are full on aerobic. But the heterocysts, they're like, nope, no oxygen. Let's just devote this to fixing nitrogen because nitrogen has to be in an anaerobic. To fix nitrogen, the enzyme needs to be in an anaerobic environment. And if that enzyme gets exposed to too much oxygen, it just kind of falls apart and stops working. So that's a cool option. Then there's fruiting bodies, which are so cool. So this is actually a bacteria, Myxococcus xanthus, that often just has a relatively normal lifestyle, unicellular clusters of microbes hanging out. But then if things get tough in terms of usually lack of nutrients, then they start to come together. So they actually do quorum sensing for this too, and they'll start to find each other and gather. And now you're starting to see after 7 hours, they've kind of created some kind of structure, and then it starts to become a little bit more obvious as the time goes on that you get this mound of cells that have come together. And the reason that they're doing this is that they're under usually starvation conditions. And so they will actually create a situation where they come together. If they still maintain, there's, no nutrients, then what they do is they'll form spores. At least some of the cells will form spores in that fruiting body, just so that some of the cells can persist through the tough time. Many of these cells will die off, but the ones that are forming spores will survive. So let me show you another picture here. And so most of this mound here, these are all spores. Okay, the bottom part that's sticking to the substrate are just the regular cells, but most of the mound is spores. And then here's a drawing where they're trying to show, how they aggregate and then they form this fruiting body. So it has this differentiation. Here you have the part that's sticking to the substrate, and then often you have some kind of stalk that's made out of cells, and then the spores will form. And so it's a way to differentiate in this part of their phase of their life cycle so that they can create spores that will persist. So at least some of the cells make it in the spore state until hopefully time gets, things get better for them. So here's some nice images of different myxobacteria. Now these are tiny, tiny, tiny. Okay, these are bacterial fruiting bodies. So they're nothing like a fungal mushroom that you see. They're tiny. But I thought, wow, these are great images that were taken. So, and they're so cute. Too bad they weren't more visible, but you definitely need microscopes to see this. But you can see quite ornate design. You can see why they call it a fruiting body. I mean, it does really have this differentiation of different cells, have different roles to play. And then you have a nice spore production going on. Okay, so
And we'll talk a lot more about fungi after break, and their fruiting bodies, but that's a separate topic. The Streptomyces bacteria are also capable of differentiating in their life cycle, depending on the conditions around them. So the word streptomyces, myces is referring to something fungus-like, so that unfortunately that name stuck, even though they figured out finally, oh, they're not fungi, they're bacteria. Strepto means bent or twisted. So if you look at that first image, figure A, you can see it looks like bent or twisted mycelia, which we actually use that word for fungi, but we use it here too. So these are hyphae or mycelia that are growing, so long thread-like growth form. And then if times get tough and they're like, ooh, I'm running out of nutrients, they start to form aerial hyphae. So if you grow them on an auger plate, here are colonies, figure B, colonies on an auger plate. And all that kind of gray that you see, that's aerial hyphae coming up off the auger. And then when they start to change color, that's not Photoshop, that's its real color. And Streptomyces can be different colors, it depends on the species. But usually visually, when you see that color shift from gray or white powder to this blue, in this case, blue powder, then they're forming spores. And they will coincide. When they're forming spores, they'll pump out antibiotics. And so sometimes you might see a colony on a plate that has these like crystallized, looks like liquid crystals, that have crystallized. And that's actually probably pure antibiotic that's sitting on a colony. So the bacteria have exuded that antibiotic to such a high level that it oozes out, and then when it hits the air, it starts to kind of crystallize. So that's like liquid antibiotic that's crystallized. So you can see in figure C all the spores that are forming down the length of the aerial hyphae. So again, this idea of differentiation through, they have a more complicated life cycle and can differentiate themselves.
Why do bacteria make antibiotics for other bacteria?
Oh boy, that has been a question that has been debated over for decades. It's been really interesting to see over time, how people have debated about this in published literature. First, they thought it was just so one bacteria could kill another bacteria. And that they think that still can be going on. But now they're also realizing antibiotics, like when you look at the antibiotic chemical structure and start looking at the physiological pathways at low concentrations, which most soil bacteria, like they'll put out antibiotics, but it's usually at fairly low concentrations. Like, when you look at it, it's like, well, that's not really enough to kill. So why are they doing that? And so they're finding that the ones that live in a more natural environment, they're pumping out small bits of antibiotic to communicate. It's actually a communication tool instead of a killing tool. So that when that came out, that kind of people went, what? But it makes sense because there's all kinds of natural systems where there's just a little bit of antibiotic coming out, and it didn't make sense to us. We're like, why would they do that? Because it's expensive. When you look at the physiological pathway to make an antibiotic, it's costly. And so we think now that there's, they can use antibiotics to kill, but they can also use it to signal and communicate. So the saga goes on. It's a saga. I mean, people are like debating and fighting about this. Sometimes it's very fun to read those. But anyways, so very interesting group of bacteria that can actually, they look like little petite fungi under the microscope with all those mycelia growth forms. Here's just a drawing trying to show the complicated life cycle. I love working with them because you've worked in the micro lab now enough, half the semester. Do you like the smell of most of those bacteria? Try teaching labs when you're pregnant and have to smell that stuff. Oh my gosh, when I was pregnant with my boys, I had to teach labs, and when you're pregnant, smells can really get to you. My biggest fear was I was gonna puke in front of my students, and I never did, but came close a couple of times. But anyways, they smell like the smell of wet dirt after a rain, and I love that smell. So when you walk into my lab, my research lab, if I'm working with a lot of Streptomyces, it smells like a nice rain just came through and wet the soil. It's lovely. But anyways, you can see that they have this, substrate form of growth, and then they start to pop up off the substrate. And then as they start to segment into spores, that's when they kick out some antibiotic. So, and then the spores get released and they go at it again. So this is just kind of a summary figure, just showing some of what we talked about. And so, the idea that bacteria can exhibit kind of a multicellular form, almost a tissue-like form sometimes, has really intrigued a lot of scientists, especially those that are looking at the evolutionary process and, how did we go from prokaryotic cells to eukaryotic cells to single cell to tissue-like cells and multicellularity. A lot of interesting research going on with that. So microbes can be very cool when you start looking at some of these more extravagant ways that they live their lives. It's not just all let's, do binary fission and call it good. They can have a very complicated way of going through their life cycle. Okay. Well, I guess we got to stop there. And so the next topic will start, what is today? Wednesday? Oh, Friday. So we'll start quorum sensing on Friday.